All right, so we have been talking about the different types of media. We talked about the fact that we can classify media based on its physical state, its chemical composition, or its functional type, like what do we want it to do. And so far we've just talked about the physical state of the media. And we said it could be solid, semi-solid, or liquid media. So let's move on and talk about the chemical composition of the media. So the chemical content of media can be chemically defined, or it can be what we call a complex media. So the chemically defined media has a very defined list of ingredients, and these ingredients are measured out very precisely, and all ingredients are basically known. Right, so we know exactly what is in that media, and we are using very specified amounts of each one of those ingredients. If we contrast that against a complex media, so complex media has at least one or more components that is not chemically defined. In other words, we don't know exactly how much of that component might be present. So we have examples of ingredients that we might find in a complex media as that being products from plants or yeast or animal. Uh, in lab, a lot of times we use what are called blood agar plates. So blood has been introduced into the media. A lot of times it's horse blood or rabbit blood. And we consider this to be a complex media because every blood sample does not necessarily have the exact same chemistry. It depends on the health of the animal. So we may not know exactly kind of all the different um, types of molecules that might be in that particular blood that was used in that particular media at that time. So we call that complex. I just wanted to give you an example of a defined media just to kind of give you an idea. So it's when you make these media, it's kind of like following a recipe. So here we have like 0.25 grams of each of these different amino acids. You have 0.5 grams of other amino acids, 0.12 grams of some other ones, and then you've got some vitamins that you're measuring out in moles. Um, and you're bringing it all up to a certain volume when you're making this media, right? So once again, defined, it's very defined. We know exactly how much of each one of these different amino acids is present in the media versus a complex media where we don't know exactly how much of each of these amino acids might be present. Just depends on the microorganism, or excuse me, just depends on the organism from which we've taken um, that component from. All right, so that's our chemical composition. Now let's talk about the functional type. All right, we said what is the job of the media? Kind of what role do we want it to accomplish? So we have what we call general purpose media. And so this is basically media that's not special. It basically works well when you don't know what you're looking for. So you're just going to kind of swab your inoculum on it and kind of see what pops up. Usually all microorganisms kind of look the same on general purpose media, but it's typically a way to grow something initially, just to get it to grow, to see what you've got, and then you can investigate it further. We have enriched media, which helps to grow fastidious organisms. So that word fastidious means they require certain nutrients, right? They're very fastidious in their requirements, right? So they'll only grow if certain ingredients are present. So an enriched media, think of it as it's been enriched with something. So it could have blood, it could have different growth factors, all right, that are required for the growth of these microorganisms. So we're kind of giving them exactly what they need, all right? So as it says, it's used a lot of times to encourage growth of pathogens that are usually present in very low numbers, and so they're typically kind of a little bit more difficult to grow, so we use an enriched media for them. Then we have what we call selective media. So selective media allows us to select for certain microorganisms, right? So as it says, it contains one or more agents that inhibits the growth of a certain microbe. So it's going to prevent one microorganism from growing while allowing another microorganism to grow. Right, so as it says, it's important the primary isolation of a specific microorganisms from a sample when you have different species. So it keeps certain species from growing while allowing others to grow. All right, so as it says, it kind of helps us get rid of our unwanted background organisms. So we could use an antibiotic in a media, and therefore only the organisms that are resistant to that microbiotic, or excuse me, resistant to that antibiotic will be able to grow, and those that are not will be killed off. They won't be able to grow. So that is selecting for the antibiotic resistant. We could be using salt in our media, and therefore only the microorganisms that are able to tolerate salt will be able to grow, and those that cannot will not grow. So once again, we're selecting for certain microorganisms. A differential media allows us to grow different microorganisms on the same plate, but we can view or visibly see the differences in these microorganisms. So as it says, it allows multiple types of microorganisms to grow, and we can see the differences in them. So on this plate, this solid agar plate that we see on the right, it was actually, the media originally was pink, like you see in the top portion, and we have two different species of bacteria that are growing on it, and one has turned the bottom half of the plate yellow. So the bacteria that's growing on the bottom half has changed the color of the plate. So this allows us to differentiate between the two different bacteria. So because of the differences, we can physically tell the differences between them, right? So sometimes it's the colony itself that might change color on a differential media. It could be that different gases are being detected in a differential media as well. All right, so it allows us to differentiate between the microorganisms that are growing on the plate. So here's an example where we've got some broths, or broth tubes, and it says it's a mixed sample. So we have a couple of different microorganisms that are growing in our broth tube. Now, we see we're going to take an inoculum from this broth, and we're going to plate it out onto our solid media. If we use a general purpose media, we see that all the microorganisms are growing. If we plate that same inoculum onto a selective media, we're not going to see all the different microorganisms that were in this original sample. Only certain ones will be able to grow. So in this case, they're showing just one species that is capable of growing versus all the different ones that were able to grow on our general purpose. So once again, general purpose is one that we use to kind of initially culture our microorganisms to see what's there. Selective allows us to select for certain microorganisms and prevent the growth of others. If we look at our broth tube again, in this case we still have our general purpose media, but now we're going to use a differential medium. So the differential allows all of the bacteria to grow, so all the different species that we might have on our multi-purpose plate are still able to grow on our differential media, but you see that there's a lot of different colors to the colonies now. So we have kind of this bluish purple colony, we got a red, we got a white colony, and this allows us to differentiate between the different species. So that's our differential media. So some media can be selective and differential at the same time. So they can be both selective and differential. So we have the example of a MacConkey agar, which, as it says, will suppress the growth of some of the organisms, and then those that are able to grow, we're able to see visual differences between them so that we can distinguish between the different species that are able to grow. So we can prevent the growth of some microorganisms, and those that do grow, we can distinguish amongst them. Now, with these differential media, a lot of times we're using dyes to help us see the differences, and these dyes are usually natural pH indicators that cause a color change within the colony or a color change within the media. So a pH indicator allows us to detect a change in the pH. And so that means that the metabolism of the bacteria is usually producing some acid or producing a base that is a byproduct of its metabolism. And that pH indicator shows us a change, that the pH has either dropped or the pH has increased from what it was. And that allows us to gain some information about that microorganism. Let's say that it is a lactose fermenter. It's able to break down lactose or break down other carbohydrates and produce acids. And because it's making that acid, we're able to detect it with the pH indicator or the dye that is present. And that then allows us to differentiate between the microorganisms that are growing. We have a few other types of media. We have what's called a reducing media. So typically this is used for anaerobic organisms, so organisms that do not grow well in the presence of oxygen. So thioglycolate broth is an example of a reducing media where it basically helps to kind of slow the penetration of oxygen through that type of media, or helps to bind up the oxygen so it's not present for the organism. We have transport media. So as it says, it's used to maintain and preserve specimens as they're being transported to a lab, right? So not necessarily allowing the microorganisms to grow, but simply maintaining them. We don't want them to die before they get to the lab, and we don't want other organisms to outgrow other ones that might be present within that specimen. We just want to preserve what's there so that it can get to the lab in time, and then they can investigate it. We've got carbohydrate fermentation media. So we were just talking about microorganisms being able to ferment or metabolize certain carbohydrates, and then we can see changes in the media. So this is showing going from alkaline or a base to an acid that could be produced. So you see a change if it's able to ferment the carbohydrate. And then, as we mentioned, being able to see gas that might be produced. So there's actually little tubes that are in this broth media, and they're inverted upside down. And if gas is produced, it gets trapped at the end of the tube. And so you see a little bubble there that shows that it has produced gas. There's also assay media, right? So assay media is used for testing the effectiveness of certain drugs or disinfectants. So how well does an antibiotic work against a particular bacteria, or how well, let's say, does Lysol work against a particular bacteria? There's also what's called enumeration media, and it's used